So without any further ado, I'm delighted to introduce you to our speaker this afternoon, who is Dr. Antonio Zanotti. Antonio has been at Johnson Matthey since 2003 and is now R&D Director of the Catalyst team in Cambridge, UK. And he's going to talk to us today about homogeneous ester hydrogenation. So I'll hand over to you, Antonio.
Okay, hello to everyone. Thank you very much for joining us. As Claire said, the subject is homogeneous ester hydrogenation. It is something that the group here feels quite excited about. It's a new technology. I hope that you'll find something interesting in the webinar, and I hope I will manage to convey some of the excitement that we feel about it. So what we're going to discuss today is the reduction of esters to primary alcohols. Those can be aliphatic or aromatic esters. The area has been widely reviewed, so I will put as many references as I could fit in the slides for you to go back and look up if you find anything interesting. But fundamentally, this talk is not an academic talk. So I will basically mention no mechanism or almost no mechanism. And instead, I will try to focus on what we feel are the interesting things and these, sort of applicative details that will lead, we all hope, to real industrial applications of this technology. So the agenda today is a very brief introduction about why we feel this area is important and why we feel that it will have an impact in the next few years. There will be a little bit of an historical overview with apologies to the many contributors to the areas that I had to leave out. And throughout the talk, I will try to point out an industrial perspective, the things that do count in terms of making this technology relevant to industry, and of course the potential for disruption and the potential for applications. So what we expect from ester hydrogenation is, of course, to make primary alcohols, but from a variety of substrates. There is a vast array of challenges facing this generic umbrella of reducing esters to primary alcohols, because of course when you work in the life science space, you never really use a simple substrates, but they're always complex molecules with a lot of heteroatoms, functional groups that can also be reduced or hydrogenolized. Particularly important is the selectivity of ester reduction versus CC double bonds, which is, of course, relatively easy to reduce as a functional group. And then there is the issue of chiral substrates. It's important to avoid racemization of the existing esterogenic centers. And then we often deal with diesters. One can think of using also this technology as a deprotection route. So there are really a number of challenges that go beyond the simple substrates that are usually the publication, which are usually published by academics. But even more interesting about ester hydrogenation is exploiting the reversibility of the catalyst, because this catalyst they can reduce esters. They can also do the opposite. They can take hydrogen out of primary alcohols, either to make another ester or to make a carbonyl, which can be trapped in situ by something else, an amine, for instance. And this leads to the construction of molecules such as amines or amides or alpha alkylations. This is a new area, which is often referred to as hydrogen borrowing. It does really has in itself quite a lot of opportunities. Think simply of the possibility of . Of getting away with alkylating targets, but using instead alcohols. I will not focus on these new areas, but throughout the talk there will be references to some applications where ester hydrogenation catalysts are used for this new type of transformations. And of course, the beauty of it is that we can do really new chemistry using catalysts that we understand relatively well, that we can make. So again, this adds to the potential. Whenever there is an emerging new technology, really you need two things. You need a push and a pull. A pull from the need to make targets, in this case primary alcohols, and the push. So there has to be behind the new technology an evolution that makes for the technology possible to deliver what it offers. In this case, I would like to point out that homogeneous hydrogenation in its chiral version, in its asymmetric version, has been around for, well, at least 40 years, has been widely used in industry, is widely accepted, but it is only in the past probably 20, but even only 10 years, that there has been a definite move of interest in the R&D area from chiral application to achiral applications. This requires much higher turnover numbers from the catalyst, much higher efficiency, because of course you don't have the privilege anymore of working on chiral targets that can take quite a relatively high cost contribution of the catalyst, but the transformation must be much more cost effective and efficient. There are various routes, of course, to primary alcohols. They can come from reduction of aldehydes, they can come from reduction of esters and acids. These two classes of substrate might have different origins. Maybe aldehydes are more from the oxo process, so from petrochemicals, esters a bit more from biomass. What I say about esters, of course, can be applied also to aldehydes. So the catalyst that I'm going to discuss do reduce carbonyl bonds very efficiently. But the interest is, of course, on the much more difficult target of esters. Acids are a little bit more of a difficult area for the moment, although there are already some indications that the catalyst can be extended to operate on acids too. So what is the competing technologies? There are basically two well-established routes to reduce esters. Larger scale applications mostly are based on fixed bed catalyst, copper-based, other metals. They are very efficient, very green, of course, because this is heterogeneous catalysis. But of course, they do operate under harsh conditions, relatively high pressure, certainly relatively high temperature, and basically they do apply mostly to larger scale products. Much more flexible and applicable to the complex molecules of life science are hydrides. These are very well understood reagents. People have been using them for a long time, and they have some fundamental drawbacks. And I think that it is these are the main comparison that we have to do. As homogeneous ester hydrogenation has got the potential to remove or bypass some of the limitations that are associated with the use of hydrides. In order to define the scope of homogeneous ester hydrogenation, there are various ways that one can proceed. And this is just a snapshot of some of the work that you've done. You can sort of look top down and consider all the applications for hydrides, which are in the literature. About a quarter are for the reduction of carbonyls, another quarter is for reduction of esters and acids, and then there is the reduction of CN double bonds, which is very useful. but homogeneous catalysis is still in its infancy in this area. So if you look at this graph, basically you find that there is about half of the graph where ester hydrogenation catalyst can be applied. Or you can build your model a little bit more bottom up. So basically you look at all the primary alcohols which are in the literature, which are industrial targets of interest. And actually interesting, it is interesting to see that the pharma pipeline or the pharma drugs are not necessarily the biggest area in terms of volumes and values for primary alcohols, but it is instead the FNF industry that has got in mind some of the highest value primary alcohol targets. And you will see, in fact, that a lot of the research in the area of ester hydrogenation has been driven by the FNF industry. There have been real innovators in this field. Well, the fundamental metric that you have to consider is always cost when you talk about industrial applications. Hydrides per se, per kilo, are cheap. I mean, people might disagree on these numbers, particularly on the estimated cost contribution. This is what we sort of estimated to be the cost of obtaining a kilo of product using certain hydrides. It's just a ballpark figure, and it is still important for anyone working in this area to compare our new technology with the established one. And to be honest, cost contribution is the fundamental parameter, probably the first one to tackle. We have looked at these numbers quite a lot, and we are pretty much convinced that once a decent turnover numbers is achieved using ester homogeneous catalyst, the cost contribution of hydrides is matched and improved. So per se, the fact that per kilo hydrides appear to be a lot cheaper than any homogeneous catalyst is not the main hurdle. You need also to take into account that there are other things to be considered: the efficiency, the sustainability of the process. There are lots of metrics that can be applied to that: the PMI, the E factor. Again, we played a little bit of the game of comparing our experience in using homogeneous catalyst with what has been published. Let's say we choose OPRD because it reports quite detailed procedures. And these numbers fundamentally just put in numbers what everyone knows. The application and workup and waste generated by the use of hydrides is massive, and it poses a massive problem when compared to the simplicity of an hydrogenation process. Of course, heterogeneous catalysts are even more green in a sense than homogeneous catalysts because of their recyclability. But again, I think that it's much fairer to compare like for like, and hydrides are the main challenge to tackle for our technology of choice, the homogeneous ester hydrogenation. So I think that the catalysis approach can provide very cost-effective processes at low loadings, very high chemoselectivity, cleaner and safer workup, and fundamentally more controllable reactions. The real problem is that the hydride reductions are well understood, and there is, of course, an understandable resistance to look into the application of new technology, which also requires specialized equipment because it requires to operate under pressure. Also, we will see that there is no real requirement for the use of high pressure. and pressure below 10 bar can easily be applied. In the overall, beyond the cost, I think it is fair to say that the environmental and safety advantages of using hydrogenation technology does really bring cost benefits to the application on real targets. Ester hydrogenation technology can be seen a little bit as an evolution of carbonyl hydrogenation. In the 80s, the first homogeneous catalyst to asymmetrically reduce ketones were based on phosphines and ruthenium. Binary ruthenium was introduced by Noyori in the second half of the 80s. The substrate that you can see, not of the ester functionality, which is there only to chelate the ruthenium. So this catalyst work around a so-called inner sphere mechanism, and this is my only mention of the mechanism throughout the talk. The ketone has to be close enough to the ruthenium hydride, and the ancillary functionality of the ester is only there to provide this bidentate chelation to the ruthenium. The second breakthrough in this area came again by Noyori, who in fact got a Nobel Prize in 2000 for these innovations and many others. And in the mid-90s, he demonstrated that you can actually reduce non-functionalized ketones by modifying the catalyst in a way that the ruthenium becomes saturated with 6 coordination sites, which are filled by phosphorus and hydrides and nitrogen. But fundamentally in the structure is the presence of an NH2 or NH unit, which basically forms with the ruthenium hydride the scaffold on which the carbonyl approaches the catalyst and is reduced. This is called an outer sphere mechanism because the substrate which is reduced, it never really directly coordinates with ruthenium. There is a number of, there are a number of publications that actually debate the real nature of this mechanism. I mention only 2 of the very recent ones, and there are various possibilities of alkoxide directly coordinating to ruthenium. But what remains common to all these catalysts is the presence of the NH functionality in the ligand scaffold. And when the catalyst started to transition from ketones to the more difficult targets, esters, this functionality, this particular feature of the scaffold, the presence of NH, stayed there because it is fundamental to achieve high activity. Together with a plus, with the advantages of this concept, comes also a disadvantage, which is all these catalysts based and working on the Noyori-type mechanism, they require activation with a strong base. Not only activation, but usually base is required throughout the catalytic cycle. And we will see that this is actually something that sometimes causes a problem in ester reduction. So this is my naive tentative to give a bit of a timeline of the evolution of ester hydrogenation, homogeneous ester hydrogenation. At the beginning was in the late 90s, a tridentate phosphine called Triphos. Triphos was initially reported by Elsevier and others, forming in situ a catalyst with ruthenium macaque. They demonstrated the reduction of a few activated esters, diesters, fluorinated esters. These catalysts work with an inner sphere mechanism. So in a sense, they are just a derivation of this first generation of ruthenium phosphine catalyst that I mentioned in an earlier slide. Then, of course, people find that there is an advantage in making preformed catalyst. So various preformed ruthenium-2 catalysts have been isolated and employed bearing the Triphos ligand. It started a sort of with relatively few papers in the late 90s and then 2000s. And then the literature about the use of Triphos literally exploded in the last few years. So it is a bit of a rhetorical question that I posed in the slide. It's a privileged ligand. Yes, it is. If you simply judge by the growing number of applications, also with different metals, ruthenium, cobalt, iron, manganese, and it can be used in a number of applications which go well beyond the reduction of esters. People have demonstrated depolymerizations, reduction of CO2. It is one of the few catalysts that allows the efficient reduction of amides to amines and so on. We have ourselves played a little bit with a preformed Triphos catalyst, demonstrating very high activity in the reduction of aldehydes, in reductive amination, to making amines. And others have been working on other interesting transformations, such as going directly from an ester to an ether without the intermediacy of the alcohol, which is a transformation which can be of use. The second step in our little ride across the history of homogeneous ester hydrogenation is the switch from the inner sphere catalyst to the outer sphere catalyst. So basically what happened at some point, various researchers who were well used to utilize Noyori-type catalyst in carbon ion reductions managed to find a trick to use this type of catalyst for the more challenging target of ester hydrogenation. And this has happened at the same time by various groups. One very important contribution that I'm not going to expand on is the work by Milstein. You can look it up. There are plenty of reviews. Milstein catalyst work along a sort of a similar mechanism as the one employed by Noyori-type catalyst. Our experience is that they are not the most efficient ones in ester hydrogenation, although there have been recent papers that show improvements, and of course they can really be used in a number of very important and challenging transformations. So my apologies to Professor Milstein. I'll sort of stick with my history of Noyori-type catalyst. And as I said at the beginning, really, the first examples were even using chiral catalyst. What these authors did was to find a way to utilize Noyori-type catalyst on esters and lactones. And fundamentally, the trick consisted in the switch from alcohols to aprotic solvents and basically using more base. The biggest contribution in this area came in 2007 from Firmenich, so an FNF company. They basically had chiral versions of the two catalyst family reported here. They made some achiral versions of these catalysts, and they first demonstrated really good two-nova numbers on a vast array of substrates, again using aprotic solvents and a relatively high amount of base. You can see that certain CC double bonds are still challenging for this catalyst, particularly if they are a bit activated, such as in cinnamic ester, or they are terminal CC double bonds, but there is also very good selectivity as soon as a double bond is substituted. And these are very good catalysts. The idea is, keeps being used. There are recent publications of manganese catalyst pairing this type of PN ligands. People have demonstrated that you can do a metathesis of long chain esters and then add a PN ligand, and this modifies other PIs in situ the ruthenium catalyst, which becomes an ester hydrogenation catalyst. The next step was to move on from scaffolds and engineering of the catalyst, which was fundamentally based on the experience of chiral catalysts, to more purpose-designed catalyst for ester hydrogenation. Same concept of an outer sphere mechanism, but the tridentate ligands, so more robust and purely dedicated from the beginning to achiral reductions. In 2012, 10-12, researchers at Takasago introduced the first widely commercialized catalyst, ruthenium macho, based on the tridentate scaffold, PNP ligand. And maybe the most interesting application was the demonstration of the reduction of lactate, so a rather difficult substrate that requires either the use of a pre-formed hydride catalyst or a very tight control of the reaction conditions in order to avoid arsonization. And of course, whenever you are working on chiral substrates, it is important to minimize the use of base, usually to keep it low with the temperature. And of course, this can be compensated by the use of a higher pressure. So the PNP ligand turns out to be widely applicable to a number of different applications, metals, ruthenium, osmium, iron, manganese. This is a bit of a just visual, attempt to describe the variety of transformations that can be enabled by this catalyst. I would like to, to express my opinion that probably all tridentate catalysts can do this kind of applications, and it is probably the next step for the R&D community to expand the use of tridentate catalysts. Certainly, you can replace phosphine ligands with carbines. This is well known. Again, I mention only some of the structures that have been introduced by Milstein, Pitko, Sanchez. And one particular application, again, coming from the same group that worked on macho, where they added a carbene in situ, and basically they transformed the ruthenium macho catalyst by replacement of the CO with a carbene. And the catalyst became more active at very low pressure of hydrogen. When you actually look at the numbers and the details of the paper, this might not necessarily be the way forward to have a very efficient transformation, because ultimately the paramount thing to do is always to increase the turnover numbers. But of course, it does point to the fact that pressure is an important parameter to be considered in applications, and there is a desire to operate as hydrogen pressure as low as possible. Few years after ruthenium macho, Professor Dimitri Gusev from the University of Toronto in Canada introduced a number of different tridentate ligands. He explored the scope of PNN, SNS, NNNP ligands with, again, different metals. He has published various times these innovations. Around 2015, we tested this catalyst. We compared with the catalysts that were available at the time. And again, this is a little bit of a visual thing. We certainly demonstrated at least for our own purposes, that Gusev catalysts were up there in terms of efficiency with the best catalyst available on the market at the time. The pyridine NP structure doesn't only do ester to alcohol reductions, it can do the reverse reaction, for example, ethanol to ethyl acetate, and can reduce imines to amines. The SNS ruthenium system has been reported to reduce a number of esters and carbonides. We are, experimenting a lot with this system, as I will show. You can see that the turnover numbers, or you can express them as a substrate to catalyst ratio, increase throughout this historical ride through ester hydrogenation. And also, what is increasing progressively is the selectivity, ester versus the CC double bond. And if anyone is interested in the mechanism, I have added a reference at the bottom of this page. And then equally, the osmium catalyst is very versatile. Again, you can see an increase in turnover numbers, an increase in selectivity, the use of a milder base, and interesting reactions, again, alcohol to ester, alcohol to amide, and of course, ester reduction. And then maybe someone can tell me how they feel about the use of osmium catalysts. This paper is interesting, or was interesting when we found it, because it gives an industrial, practical perspective on what people want from an ester hydrogenation catalyst. They clearly state that the aim for their testing was to show, to find out whether the catalyst could be used at low loadings, if it was sort of, in brackets, cheap, operationally simple, and if it could be used under reaction conditions which were robust and not too high pressure. The conclusion was sort of yes, although the turnover numbers that they were reported in the paper were actually far lower than what we usually find. So optimization in reality takes you a long way. Another interesting consideration was that they tested other structures and not all of them were active. So in reality, not everything in the space of the tridentate ligands leads to active catalysts. We have demonstrated with ruthenium SNS, again, the reduction of lactate, similar to ruthenium macho. We had to change the solvents from methanol to ethanol. It made a big difference, and as always with homogeneous catalysis, it points to the fact that yes, we can predict some general reaction conditions, but then each catalyst requires its own specific optimization to make the most out of it. We have demonstrated the use of ruthenium SNS catalyst with very good activities on methyl and ethyl oleate, technical grade, neat, low loadings. Again, this has been the subject of studies by other groups. See the reference at the bottom of the slide. And there was a time that in the lab we had some fun hydrogenating olive oil from the supermarket. It's actually an interesting substrate because it requires to achieve some good ester to CC double bond selectivity. The triglyceride itself is a tougher substrate than the isolated methyl or ethyl ester, and it works neat, which is often a requirement for many larger scale applications. And you can see the temperature dependence, particularly in the case of the SNS ruthenium catalyst. So the blue bar is the saturated product, the red one is the unsaturated one, and as you decrease the temperature, the selectivity increases as well. The space of the tridentate ligand is not limited to the structures published by Gusev and ruthenium mature. There are other possibilities, and other groups have been working in parallel on conceptually similar catalysts. I just mentioned the work by DSM and Givaudan, because of course they have generated some very efficient and industrially applicable catalysts. Givaudan have demonstrated published the reduction of lactone to sclerodiol, which is a precursor to an important FNF target. De Vries et al. And others have demonstrated a very high activity and possibly the best selectivity so far of the reduction of ester versus the CC double bond on a really difficult substrate such as the cinnamic ester. And of course, then they went on demonstrating aldehyde hydrogenation, gamma valerolactone hydrogenation, neat, and so on. The last step in our historical overview is the introduction of the tetradentate ligands. They certainly further increase on the activity. They also introduce a degree of complexity that probably goes a little bit beyond the complexity of the tridentate ligands. So the jury is really out there to define at some point which one of the many catalysts which are coming up more and more frequently in the literature are going to be the ones that that really produce efficient industrial processes. But the numbers again are very nice with this work from Zhu, from Zhang. Again, you can see increasing turnover numbers and increasing selectivity towards the ester reduction. The last part of the talk is focused on a couple of points which I think are important, because all these catalysts, they look very good when published, but then it's the next step to take them into real applications. So there are always a number of difficulties that have to be overcome, and that really make the difference between having an academic result and having an industrial process. I already mentioned the impact of volume efficiency, so working either neat or very highly concentrated, but there are other parameters that make a difference in terms of feasibility of a reaction. Temperature, pressure. Can we do transfer hydrogenation instead of hydrogenation? And then basically the substrate purity. So the feedstock purity is always a fundamental point in all homogeneous catalysis. It's often has been historically a killer for very good reactions. Of course, it's very hard and a case-by-case situation to define what purity, what impurities count in terms of impacting the catalytic cycle, and we only focused on briefly on the impact of water in the substrate. Temperature. When you use the very first generation catalyst based on Triphos-Ruthenium, you'll find that operating temperatures tend to be well above the 100 degrees. Usually you require 130, 150 degrees, which means that it is relatively difficult to achieve good selectivity when in the presence of other functional groups that can be reduced. I present here a little study that we did comparing an SNS and a PNN ruthenium catalyst. Of course, it's easy under other conditions to achieve full conversion at 20,000 to 1. But in this specific case, we wanted to achieve a partial conversion in order to compare the two catalysts. And the results is actually quite indicative of what happens because the ruthenium SNS catalyst is much more efficient towards the lower range of temperatures, and then the situation becomes the opposite with the PNN ruthenium catalyst. And then when you go too high with temperature, it performs very well. So again, this goes in the direction of it is important that each catalyst is optimized to find the right set of operational conditions. Pressure is important because it is quite useful to achieve high turnover numbers, to use high pressure, but for a number of applications, it's also useful to be operating at a low pressure. And basically the trade-off is between pressure and the catalyst loadings, not between pressure and temperature. As you've seen in the previous slides, trying to compensate for lower pressure by increasing the temperature is probably the wrong way to go. You basically need to use more catalyst, but that doesn't mean that the reaction becomes not viable, because still at the 5 bar, it's possible to achieve full conversion at quite a good catalyst loadings. In a sense, it would be even more interesting to be able to avoid the use of hydrogen and to use something like formic acid, which is a classic transfer hydrogenation reagent in the case of carbonyl reduction, but this hasn't been demonstrated to be possible yet with ester hydrogenation catalyst. There are only a couple of papers that fundamentally use a metathesis, an ester metathesis reaction. There is a sacrificial alcohol which is oxidized to the corresponding ester, and then the ester goes to the primary alcohol. If you don't have an excess of one of the two reagents, as you can see on the left-hand side, you basically get a mix of all the possible scrambling products. Water, as I said, again, a bit of a curiosity. At some point, we were interested in defining what happens when you do not know how much water you have in your substrate, or if you add a certain amount of water. And in a series of experiments, we found that the addition of 1% volume of water basically completely shut down the reactivity of the 3 ester hydrogenation catalysts that we tested. It can still be compensated by adding more base. So there are ways around it, but it is a parameter that we recommend that people should keep in mind. Of course, where they work on esters, these catalysts, they can work very efficiently and very easily on ketones and aldehydes, and you can see here that the turnover numbers are very, very good and very simple optimization. But the interesting slide is this one, because when we tested the use of water on aldehyde reduction with the same catalyst, we found a trend which was completely opposite to the one seen for esters. So while the presence of a few volumes of water shut down the reactivity on esters, it becomes instead fundamental to achieve high conversion on certain unsaturated aldehydes, such as furfural and cinnamaldehyde. I preempted a question on the why. I'm really not sure. So it's basically, I don't know. Probably we are playing with the overall basicity or pH of the reaction, and this is somehow correlated to the pKa of the intermediates of your reaction and the stability of the ruthenium hydrides. Last 2 slides, I go back to the concept of going beyond the test substrates of literature. And to support this concept, I give a little picture of when we looked into primary alcohols in the pharma pipeline. We tried to associate different, classify different primary alcohols, aliphatic, benzylic, heterocyclic, chiral, and then just to show the complexity of the substrate, there were basically a vast number of targets, at least a quarter of them, that neither fell in any of these classes or basically in all of them. So we have ongoing a little study to try to understand the compatibility of homogeneous ester hydrogenation with various functional groups. And it is quite pleasing to see on the left-hand side that there are a number of functionalities in green that we have demonstrated to be compatible with the reaction conditions generally used. So basically, we know that halides, anilines, pyridine groups, certain heteroaromatics, chlorides are tolerated by specifically the ruthenium SNS catalyst, but I'm pretty much sure that this can be extended to a number of other homogeneous catalysts. Then there are a number of functionalities in yellow that are still compatible, but they require certain optimization. Just to think that whenever you have a carboxylic acid or a phenol or an acidic proton, you have a situation where you probably need a stoichiometric base instead of substoichiometric. And then there is a limited number of functionalities which are either not compatible with the basic conditions that are used or related to the ruthenium metal, or in general, those functionalities in pink, they tend to shut down the catalysis. This was my last slide. I conclude with a comment. Certainly, homogeneous asymmetric catalysis is an established tool in industry. Probably you wouldn't have dialed into this webinar if you didn't believe that. But the new frontier is to move from chiral to achiral applications. It is not only about ester hydrogenation. There is a host of potential transformations that can make a difference. And really, the various players, they can have an impact. I think the broader R&D community can keep looking for proof of new applications because they are within reach. And then there is a role of technology companies and catalyst suppliers. These catalysts needed to become available on the quality and volumes required. And of course, there is the role of the end users, because I think there is some reticence to overcome sometimes towards the use of a new technology. But I hope that I managed to convince at least a few of you that there is a potential and advantages in embracing this. I've done. I hope that you found something interesting, and I'm happy to answer any question you may have.